So thank you for this kind introduction and for the opportunity to present this interesting paper. And before starting, I would like to clarify two terms that are associated with this topic and are often misdefined. Is the gut microbiota, that is like living microorganisms present in the defined environment biome, and the gut microbiome that includes the gut microbiota and its theater of the activity, including the microbial structural elements and external internal structural elements and nucleic acids as the integration part. The human body contains nearly the same number of the microbial cells and the human cells. In the healthy intestines, Firmicutes and the Bacteroides contribute most to the total bacteria species, 90% of the gut microbiota population, and the ratio is often considered as the relative estimate of the health or disease state. Small animal studies explore the effect of the sum of the gut microbiota originated metabolites and the components, including the primary biliary acids, indoxyl sulfatate, 3-methylamine, and oxidized choline, and the lipopolysaccharide. All aforementioned gut microbiota-derived metabolites and components are absorbed across the host gut and act as signaling molecules, leading structural, autonomic, electrical, and calcium handling, remodeling, and finally to the atrophibulation. Today, I would like to focus on the lipopolysaccharide and its correlation with the NLRP3 inflammasome on the age-induced atrophibulation. But before starting, for people who met for the first time with the term of the NLRP3 inflammasome, it is well-established signaling pathway responsible for the secretion of the inflammatory cytokines in immune cells in response of the cell damage, infection, etc. And Yao et al. For the first time discovered the activity of NLRP3 inflammasome in the atrial cardiac myocytes in patients with atrophibulation. Cardiomyocyte activation of the NLRP3 inflammasome enhanced the atrophibulation via calcium dysregulation, transcription of the potassium voltage gated channel, and the promotion of the fibrosis. Coming back to the study. The study by Zhang et al. For the first time demonstrated that the lipopolysaccharide and the induced by lipopolysaccharide excess glucose enhance atrial NLRP3 inflammasome activity, resulting in the atrial fibrosis and subsequent atrophibulation in elderly. Firstly, author analyzed the fecal microbiota from the aged and the young rats. Microbiota community was significantly separated by the aging, with decreased ratio of the Firmicutes to Bacteroides. Aged rats were more prone to the atrophibulation and had longer atrophibulation duration. To investigate the role of the gut microbiota in the age-related atrophibulation, authors performed the fecal microbiota transplantation from aged rats to the young rats and young rats to themselves for the 6 weeks. The changes in the microbiota were transplanted by the aged microbiota to the recipients and successfully changed the gut microbial ecology of young rats. The ratio of the Firmicutes to Bacteroides was also decreased in the aged microbiota recipient rats. The aged microbiota recipient rats had marked atrial fibrosis and higher inducibility and longer atrophibulation duration. Moreover, the inflammasome components were upregulated. But here I would like to underline that those results of the Firmicutes to Bacteroides ratio are in contrast what we observe in the humans, where higher Firmicutes and Bacteroides is associated with atrophibulation, and the higher Firmicutes and Bacteroides ratio is observed with the aging. But it could be explained by the difference in the gut microbiota composition between humans and the rodents. Coming back to the study. To determine the role of the lipopolysaccharide in the age-related atrial fibrillation, authors assessed the plasma lipopolysaccharide in the age microbiota recipient rats, showing increased circulating lipopolysaccharide levels. It was confirmed also in the 1,152 human plasma samples, where plasma lipopolysaccharide concentration were increased with the advancing age. As lipopolysaccharide leads to the secretion of the inflammatory cytokines, mainly by activating the expression of the toll-like receptor 4, authors assessed the expression of the toll-like receptor 4 in the different organs and found that the highest expression is in the atrial tissue. In rodents, to depend lipopolysaccharide derived atrial fibrillation promotion with the aging, authors used the lipopolysaccharide antagonist in the age microbiota recipient rats, and atrial fibrillation inducibility and duration were reduced, and furthermore, the atrial fibrosis and the expression of the NLRP3 was decreased in the atria. Although the lipopolysaccharide is produced by the outer membrane of most of the gram-negative bacteria,